The overall goal of this procedure is to visualize in vivo reporter for epidermal wound response in Drosophila embryo. This is accomplished by first collecting the developmental stage where the reporter activity can be consistently detected. The second step of the procedure is to puncture the embryo using a glass needle and microinjection apparatus. The third step is to anesthetize the embryo for visualization of the reporter. The final step is to validate the reporter localization by detecting the expression pattern of RNA probes. Ultimately, the results can show the specific activation of the epidermal wound response reporter in only the cells surrounding the site of puncture injury through confocal microscopy.
The main advantage of this technique over existing methods that involve looking at wound closure is that we can see in living embryos gene expression that's triggered by puncture wounding.
This method can help answer key questions in the tissue repair field, such as how cells surrounding an injury regulate an immediate wound response to promote repair.
The implications of this technique extend towards therapy or diagnosis of tissue regeneration because the wound response regulation is the initial step.
Wound healing can be assayed in any stage of development. However, by stage 17, the cuticle is too mature. Known wound reporters will not be activated, so to demonstrate, stage 15 to 16 will be wounded. To harvest embryos, collect 2 to